Thank you, Jose, and thanks everyone for joining us. I'm really happy that you're able to be here today. And welcome to our Introduction to the iSeq 100 webinar. In today's webinar, we will be touching on the following topics. First, we will review the iSeq 100 instrument itself and discuss some of the specifications. Then, we will touch on some of the key technologies that enable the iSeq 100 sequencing instrument. Next, we will go over workflow of both the setup and use of the instrument. We will briefly touch on a few troubleshooting steps we have found to be helpful should issues arise. And lastly, we will touch on the many resources available where you can learn more on the topics covered. I know we may have a broad audience for today's webinar, and some of us being new to next-generation sequencing and others being veterans. Since this is intended as an introduction on topics rather than a deep dive on any of them, throughout the presentation and at the end, I will be providing links to a wealth of resources. For those of you interested in more in-depth information on the iSeq, my colleague Jason will be presenting an iSeq troubleshooting webinar in April of 2020. This is the iSeq 100. It is the smallest Illumina sequencer at about 1 square foot of bench space, but it's capable of generating over 4 million reads passing filter. When run as a 2x150, it can generate over 1.2 gigabases of sequencing data. The maximum supported read length of 2x150 takes about 18.5 hours to complete sequencing, with greater than 80% of bases higher than Q30. Shorter reads will take less time and generate less data. What really contributes to the instrument's small footprint is the consumables. The reagent cartridge and flow cell contain all of the needed reagents as well as the entire fluidic system. This helps to make the setup extremely simple. Samples and reagents never leave the cartridge. The other noteworthy part of the sequencing system is the reusable test cartridge and flow cell, together known as the reusable test kit. Each iSeq 100 comes with a test kit that enables customers to install the instrument themselves, test the system whenever needed, and even allow for reinstallation if the instrument needs to be moved. Now that we have reviewed some of the basics, let's get into the sequencing by synthesis workflow and the technology used in the iSeq 100. This image represents the high-level steps involved in the next-generation sequencing process. We start with library construction, then generate clusters from the library. Those clusters are sequenced, and finally, the collected data is processed during data analysis. Before the library goes through sequencing by synthesis, it forms clusters on the flow cell. Clusters are exponentially amplified library fragments covalently bound to the oligo-land on the flow cell. Similar to the NovaSeq, the iSeq 100 uses a patterned flow cell. There are two types of flow cells, non-patterned and patterned, as used on the iSeq. The non-patterned flow cells are used on the HiSeq, the MiSeq, the HiSeq 2500, the MiSeq, the NextSeq, and the MiniSeq. The oligo-land on non-patterned flow cells is flat, randomly covered in oligos, and libraries can cluster anywhere on the flow cell. Patterned flow cells are used on the HiSeq 3000-4000X, the NovaSeq, and the iSeq. The surface of the oligo-land contains small wells called nanowells. Only a single library fragment can cluster within each well, and this allows for increased cluster density, which optimizes data output. Here is an example of a MiSeq flow cell on the left and an iSeq flow cell on the right. Within these magnifications, you can see that the surface of the non-patterned flow cell on the left is flat and library fragments can cluster anywhere. On the surface of the patterned flow cell on the right, you can see the small, neatly organized wells where single library fragments will bind. The organized distribution of wells allows for an increased cluster density by minimizing the distance between each cluster. For further information on pattern flow cell technology, please click on the computer icon to be taken to a resource. Throughout the rest of the presentation, this icon or similar icons will indicate a link. The iSeq 100 still uses sequencing by synthesis, or SBS, similar to previous Illumina sequencing platforms. SBS is the process where each base is sequenced, is incorporated one at a time, and detected before incorporating the next base. Here, I have provided a link to additional training content on our website that goes into greater detail on SBS chemistry. The new aspect of SBS chemistry for the iSeq 100 is one-channel chemistry, having two chemistry steps performed for each cycle of sequencing. Similar to two-channel technology, two images are taken for each cycle. Instead of those two images being taken in different colors, they are the same color and taken at 2 times. In between pictures, the fluorescent molecule attached to the A base is cleaved off, and a new one attaches to the C base. The T base has a fluorescent molecule that is unaffected by the chemistry cycle, and the G base does not have a fluorescent molecule attached. This leads to the instrument being able to call 4 bases from a single light source. More information can be found in the link here, and I will review even more resources at the end of the presentation. The reason pattern technology is critical here is that instead of a classical optical system that uses cameras focusing on a flow cell similar to a microscope, the iSeq 100 uses a CMOS sensor paired with the flow cell in a 1-to-1 pixel-to-cluster ratio. CMOS sensors are digital image sensors similar to those used in cameras. What is important is that they allow for the instrument to directly detect the light emitted in each cluster position with no lenses or other sensitive optical components. This image is a cross-section of the assembly with clusters in the nanowell. The last technology that enables the iSeq 100 to be a complete sequencing solution in a small box is Local Run Manager, or also called LRM. LRM is a software package that can handle user management, run tracking, secondary analysis, and allows us to view some of the results. It can be accessed directly on the instrument or remotely. What is noteworthy about LRM is that one can choose to use one or all of the available functions. Please note that we have more resources for LRM linked on this page. Next, we will be going into the workflow for the iSeq 100. In this section, the steps are outlined on the upper right hand of the slides so that we know what point we are in the process. Now we are going to be watching a video on the unboxing and setup of the iSeq 100. First, you'll want to open the box and remove all the components above the foam insert. Once the foam insert is removed, you will find your setup poster, which will walk you through all of the steps. Please remove the iSeq 100 using the 2 handles on the sides. Everything that comes with the iSeq would include the test flow cell, the test cartridge, an Ethernet cord, a power cord, a spare drip tray, and a spare filter. We will then want to open the box up, lower the front flap, and remove the top foam insert. We can remove the iSeq from the box by placing our hands by the foam inserts. Once the iSeq is on the counter, you can then remove the plastic covering, lift up the screen, remove the foam insert from behind the screen, and remove the screen protector. We'll then want to locate the power cord port and the Ethernet port on the back of the instrument. We can plug the Ethernet cable into the instrument and then the wall. We'll want to inspect the power cord, and then we can plug it into the instrument and then the wall. Next, we'll want to power on the instrument. We'll use the toggle switch on the back to turn it into the on position, and then the power button on the left-hand side will illuminate, and then we can press that to turn the instrument on. Once pressed, there will be an illumination light showing in the middle of the instrument, and then the screen will turn on. The instrument does have two different operating accounts, SBS User and SBS Admin, and you'll want to log in. The screen will then walk you through the first time setup, where we will be using the test flow cell and the test cartridge. We will insert the test flow cell into the cartridge using the grippers available on the flow cell. Insert it into the cartridge until there's an audible click. We will then want to press open door on the screen and insert the cartridge with the flow cell in and spacing upwards into the instrument. We will then choose to close the door, and once the door closes, the system checks should start. The system checks will take about 45 minutes, and once completed, you're ready to use your instrument. After the instrument is set up, this is the basic sequencing workflow. The steps are thawing, diluting the library, loading the library, setting up the run in the control software, and starting the run, and then monitoring. If you are a current user, you may notice that the thawing of the reagent cartridges for the iSeq 100 does take a bit longer than what you may be used to. It is very important to follow the thawing guidelines in the iSeq 100 system guide to allow for adequate thawing, and it is crucial for a successful run. It is also important to not go directly from dry ice to a water bath, as this can cause damage to the cartridge. As you can see on the right, the minimum thaw time is 6 hours in a water bath, and when thawed at 4C, the minimum is 36 hours. It is important to plan the runs in advance and make sure the reagents are ready to go when you are. The reason the thaw times are longer is because of the complexity of the reagent cartridge. There are reagents inside plastic tubes, surrounded by air, surrounded by the cartridge housing, surrounded by then even more air, and then the plastic bag. This necessary structure creates insulation and thus the longer thaw times. After a cartridge is thawed, your sequencing library is diluted and loaded into it. This is a simple and critical step. Successful sequencing depends on the library being fully constructed, and at the proper concentration. We always recommend adding Phi-X, which is our standard positive control. Our recommendations are for 1 to 20% spike-in, depending on the base diversity. But please consult with us here in tech support if you are unsure of how much to spike in. The system guide provides guidance on dilution and final loading. For a few library types, the system guide provides a recommended loading concentration. For others, we recommend to start with 50 picomolar and optimizing from there. The loading volume for all library types is always 20 microliters, and we will be loading double-stranded DNA. Here, we have a link to a video on how to start a run. This goes through all of the information needed to set up a run, as well as go through the difference in LRM run setup and manual mode run setup. When a run completes, the basic run metrics are displayed on the screen, showing if the run meets specifications. Once completed, no washing is required as the fluidic system is being disposed of in the cartridge. At your convenience, unload and discard the kit. You can find our safety data sheets on our website, which is linked here. Something important to note is that the flow cell is considered electronic waste and will need to be disposed of accordingly. These SDS sheets will provide the proper information used to determine the appropriate way to dispose of the reagent cartridges according to your institute and local regulations. There are three main ways to evaluate a run. On the instrument screen, which is similar to what we saw on the previous slide, in Local Run Manager, or in BaseSpace. BaseSpace is recommended for this as it will contain most of the information needed. If your lab is already using Sequencing Analysis Viewer for sequencing runs, this will also work on the iSeq 100 run folder. The instrument requires minimal maintenance. Runs will need to be deleted occasionally via process management. The local hard drive is not recommended for long-term storage, although saving locally is supported and can store a handful of runs. When connected to the instrument, the software will prompt for updates when they become available. If not connected, the software can then be updated manually. And finally, the air filters should be replaced every 6 months or more often if operating in a dusty environment. This takes about 10 seconds and does not require any tools. Now, I want to touch on some of the basic troubleshooting we in tech support use to resolve some issues that come up on the iSeq. As a reminder, this is basic troubleshooting steps, and we will be having an iSeq 100 troubleshooting webinar presented by my colleague Jason in April of 2020. There is a comprehensive troubleshooting section in the system guide. It includes this flowchart showing when to use the tools I'm about to cover. If you encounter issues, we recommend the system guide being your first source of information. Tech support's most common and most effective tool is a power cycle. It is different from a reboot in that it includes time while the instrument is completely off, allowing all subcomponents to power down completely. Whenever power cycling, don't rush and turn it back on after a few minutes. Wait 30 seconds or longer. I like to recommend using a lab timer to ensure that we're waiting that minimum 30 seconds. If you suspect that there may be a hardware issue with the iSeq 100, the self-diagnostic test called a system check can be run. This uses the same test kit as the first-time setup and is a comprehensive check of every subsystem in the instrument. If a system check passes, we know the instrument is working properly. If you ever get a system check that does not pass, please call or email tech support and we'll be able to review the logs and determine the next best steps. The reusable test kit can be used 35 times and the software will indicate how many tests are left when it is used. Another really great feature of the iSeq 100 is that it has the ability to restore to factory settings. When restoring to factory settings, only the C drive is wiped, leaving the D drive and any data on it untouched. After a restore, the control software, Local Run Manager, and any associated modules will need to be reinstalled, which takes only minutes. Every time the iSeq 100 is booted up, this screen that you see on the slide will show for 5 seconds, allowing us to choose to restore. It is normal, and you can either tap on the iSeq 100, choose to restore to factory settings, or ignore it. If you don't want to do the factory restore, ignoring it will automatically boot into the control software. If the restore to factory settings is tapped by accident, this isn't a big deal. You won't lose your data. We have found that this happens from time to time, and all you really need to do is reinstall the control software from the website and any associated modules. I've been mentioning resources throughout this presentation, and now we will explore some of what you can find on our website and how everything you need should be there. Illumina provides as many resources as possible. For the iSeq 100, this is documentation, online training courses, software, and technical bulletins. The go-to document for the iSeq 100 is the Sequencing System Guide. This has comprehensive instructions on the setup, use, maintenance, and troubleshooting of the iSeq 100. Anyone using the iSeq 100 should have this at the ready. While the software is fantastic at guiding a user through the steps, if you are newer to the iSeq or sequencing, you should have this user guide available. I personally like to bookmark it and read it in the PDF form instead of printing it out, since it does update occasionally and you want to make sure you're reading the most up-to-date information. Our online training courses can bring a person up to speed on sequencing and using the iSeq 100. I strongly recommend watching these 15 to 20-minute courses to become an expert on the iSeq or brushing up on some of the sequencing terms. The iSeq 100 Site Prep Guide has lab space and networking guidelines. This is very important when connecting the iSeq 100 to a network or domain. While the iSeq 100 runs on Windows, we don't want to treat it like other Windows PCs on an actively managed network, and the Site Prep Guide discusses this. Be sure to share this document to your IT person or system administrator before you add the iSeq 100 to your network. Earlier, I referenced the CMOS chip and one-channel SBS. This is the chemistry tech note for it, and this is the best resource for understanding the technical differences for the iSeq 100 compared to other sequencing platforms. We also have some white papers on the common applications run on the iSeq 100. If you are looking for what to do with the iSeq or want to know how others may be using the platform, these are great resources. Illumina Proactive Portal is a great tool. If we choose to connect to Illumina Proactive Portal, instrument health and performance data is sent to Illumina and can even alert us of certain issues. Here we have links for the Proactive Support page, Overview Technology Notes, and Frequently Asked Questions, which will provide us with more information about what is shared with Illumina. This is also a great tool to use for troubleshooting, as it allows us access to some of the SAV data rather than having to have you guys send it to us. It will really make things easier if we connect to the Proactive Portal. And now that we've made it through the entire presentation, we'll take some time for questions.